Welcome to our discussion on reproduction at the cellular level. In this class, we're going to briefly cover mitosis and meiosis as two different processes of cellular reproduction. Mitosis can be thought of as the process through which organisms build up the bulk of their mass, the bulk of their body. They make most of their cells through this process. So what do we mean by that? Well, organisms grow. That's one of the emergent properties of life, is growth and development. Let's look at the process of growth and development through the growth and development of the embryo of a sea star. So the sea star embryo begins as one fertilized cell. Once an egg cell is fertilized, it starts to undergo cleavage. Cleavage are events in mitotic divisions where the number of cells that make the developing embryo increase, but the overall size of the mass does not increase. So we can see here an egg cell is undergoing 2 rounds of cleavage. That first round splits it into 2, and then the next round of cleavage splits it into 4 cells. Now each one of these cells is smaller and smaller, so the overall size of the mass is the same, but there are more cells now packed in. These early stages of animal development are called blastomeres, and those are usually stages where you can actually count the number of cells. So there's a 2-cell stage blastomere, a 4-cell stage blastomere, an 8-cell stage, and that continues on until you can't tell how many cells are there. The morula is a stage of embryonic development that is solid, but composed of many cells. There's no cavity yet in that mass of cells, and that will make more sense in a moment. But essentially, the morula is an uncountable number. It's a lot of cells that make up a solid mass, and those are all the results of several different rounds of cleavage. We can see here that the mass is starting to become so dense with cells it's kind of difficult to count them. At some point, the embryo begins to develop a cavity. We call that a blastocele. Once that's happened, we call the embryo a blastula. On the bottom right, you can see that there's a cavity, a light space in the middle of that cell. Now here, it's important to note that the size of this mass has not changed from the very first cell. The only thing that's changed is that there's been a bunch of different divisions, and we've kind of divided this mass into many smaller cells. We can see on the bottom right that there's been an infolding of the outer membrane, and that's created a secondary cavity within the embryo, and this secondary cavity is open to the outside, so it's kind of like a pocket, and that pocket is surrounded by the blastocele, which is the cavity. So this is like a very early stage in animal development where you can start to see kind of how the internal tissues and organs begin to develop. And this, in certain organisms, this first kind of cavity that forms becomes the mouth, and in other organisms this becomes the anus. In biology and in science, whenever we have an organism that we look to give us an example of a process, we call that a model organism. So in this case, we're looking at the sea urchin as a model organism, and you can see that you've had these events of division. On the far left, there's one cell dividing into 2 cells. Then in the middle, you can see that those cells are divided a few times. And on the far right is a mature organism where there's been so many cell divisions, and then those cells have grown, and the overall size of the organism is now growing. So this is actually a much larger mass of cells, but it's all stemming from those initial divisions of the initial egg cell. And here we can see a bunch of cells in different stages of division. When you look at this image, it's really important that you ask yourself, what are these different shapes? What are these different structures? Are these cells? Are they different cells, different kinds of cells? And I'll tell you this, these are all the same kind of cell, but they're in different stages of growth and division. Now that you know that these are the same kind of cells at different stages of growth, your next question might be, Well, what are those stages, or what are those structures you're looking at, and why are they different? Now, before we get into kind of what's going on here, it's important that we have some information about what is needed for a cell to grow and to make different products. And what's really needed is information, directions, kind of a plan. And that plan, those instructions for making different products and for growing, those instructions are encoded on DNA. DNA is organized within the cell in a couple of different ways. Now, usually it's found in these long threads within the nucleus, but when DNA is getting ready to replicate, it starts to condense. It kind of forms a more like a organized package that makes it easier to move around the cell. We call these organized collections of DNA chromosomes. These chromosomes are organized structures of DNA and also histone proteins that help wrap that DNA in organized ways. That allows you to pack a massive amount of DNA into tiny cells. And it's stored that way unless the DNA is being used to make products, which is something we'll cover in another lecture. But on the right, you can kind of see this exploded diagram. On the very top, you have DNA. We can see that it's condensed and wrapped around these histone proteins, and those are themselves organized together and wrapped until finally we have something that looks like this familiar chromosome shape on the very bottom right, which kind of looks like an X. And we'll explain why it looks like an X. The important thing here is to note that the chromosome is kind of like a ball of yarn that is a collection of wrapped threads that are wrapped more, and those threads are wrapped more, and then finally it makes a yarn thread, and that yarn is then further wrapped to make an organized, usable piece of yarn. In our cells, we have 23 pairs of chromosomes. You can see here that we have a big list of chromosomes. One of these chromosomes actually has 3, and that's a genetic disorder that we'll talk about later. But the important thing to keep in mind here is that each one of these chromosomes occurs as a pair. Chromosome 6, chromosome 7, chromosome 8. They appear similar in a lot of different ways, but they're different in distinct ways. They're often different in the exact kinds of genetic information they have, and they might have alternate forms of the same kind of information. And so let's suppose that chromosome number 6 at the top left, on that chromosome is the gene for hair color. So one of these chromosomes will come from your genetic mother, and one of these chromosomes will come from your genetic father. You may have on one of those chromosomes from one parent the information for black hair, and on the other chromosome you might have the genetic information for blonde hair. And so depending on the interactions between those two genes, that will depend on whether or not you express the blonde hair or black hair, but both of those pieces of genetic information are available within your cells, and so they're available to be then passed on to your offspring. And we'll discuss how that might happen in the next lecture. What's important to keep in mind here is that each one of these chromosomes comes as a pair, but the exact information on each one, in this case, the way they occur in your somatic cells in your body, the exact information is not the same. They'll have slight differences. And the last analogy I'll use is that it's almost like each one of these chromosomes is a recipe for a dish, and each one of these dishes is different. So 6 might be a soup, and 7 might be a pizza. That being said, the exact ingredients for the soup for chromosome 6 might be slightly different between the two chromosomes, because you got one of those chromosomes from your genetic father, one from your genetic mother. So maybe one of those uses, like a chicken stock, and the other uses like a beef stock. Both of them have a stock, but one of them's chicken, one of them's beef. So that's kind of like an analogy that kind of illustrates how these are basically coding for the same things with variations between them. And that's really important when we start talking about selection and these other evolutionary processes. So you have within the cells in your body 23 pairs of chromosomes. They come as pairs because one's from each parent. We call these pairs, each corresponding pair, homologous chromosomes. They're homologs. The important thing to remember is that homologous chromosomes are not genetically identical. When a chromosome is getting ready to duplicate, each one of its chromatids, each one of the chromosomes from each parent, it doubles its information. And when it does that, you basically have two chromatids that have the exact same information next to them. And we call those sister chromatids. They also are attached at a certain point, and we call that a centromere. And the centromere is that attachment point of the sister chromatids. Now, the farthest point from the centromere, we call that the telomere. The centromere is important in these processes for a couple of reasons. One of those reasons is that the centromere, that area where these sister chromatids connect, is the site where you find structures like the kinetochore. The kinetochore is a point where structures called microtubules attach to the centromere, and then these copied sister chromatids are then pulled apart. A centromere can be thought of as the center of the chromosome where the kinetochore attaches and microtubules attach, and that's kind of where the copied chromatids are then split up. And that'll make more sense when we talk about this process of mitosis. But it's important to note here that there are these structures and definitions that you need to know in order to really fully understand the full depth of these reproductive processes. Let's talk a bit more about what a homolog is and what a sister chromatid is, because this is a really important point. Now, here's an example of one pair of chromosomes. So these are coding for the same genes, but they might have slightly different variations. One copy of this chromosome came from the genetic mother, and one copy of the chromosome came from the genetic father. Each individual chromosome is actually called a chromatid. When the cell prepares for growth, it copies the information on each chromatid. So here we've gone from two chromatids to two pairs of chromatids. They've doubled their genetic information, they've made copies of themselves, and now we have doubled the genetic information. Each one of these chromatids now has another one attached, and we call that the sister chromatid. Where they're attached there is called the centromere. So here's an actual photograph of chromosomes, and you can see on the right, chromosome number 1 is kind of boxed in, and that's actually not two chromatids, that's two sister chromatids. So those chromosomes have doubled, and it's not super obvious because they're so close together that each one of those appears to be an individual chromatid. But if you look carefully, you'll actually see that each one of those chromosomes is made up of a pair. That's because these chromosomes were pulled from a cell before the cell divided, and so it was preparing for that, and it's doubled its genetic information.